chapter 4, section 5, part B. Just to break it down a little bit, it was a lot. So this is the electron transport chain. It's very similar to the electron transport chain we saw in the previous photosynthesis explanations. So for right now, we're just going to look at the words. I took this picture and I made it bigger so that it would be easier for us to see. But we're going to focus on the words for now and really get down to it. So just like the last electron transport chain, the last time we in photosynthesis, we ended up taking water, we split it apart. Those electrons were charged up with sunlight in photosystem 2 to do active transport, and then in photosystem 1 they were recharged and stored in the NADP plus to become NADPH. If you don't remember all that, there's nothing wrong with you. It's a lot, and I've been looking at this for a while now, so it just kind of sticks. So this is similar in a lot of ways. The electrons are energized, they do active transport, but instead of them being stored in NADPH, at the end here, they're going to be stored in oxygen, which becomes part of water. I'm sorry, in hydrogen, which becomes part of water. Well, does it really matter if it's hydrogen or oxygen? No, it becomes part of water. That's the key here, okay? It gets stuck in the end in water. I honestly don't remember if it's oxygen or hydrogen. The hydrogens that come through are going to be stuck in water, and then the oxygens will also be in water. Now let's take a peek here. So here we have NADH. Remember, NADH was one of the Ubers we said for the electrons that came from the Krebs cycle and glycolysis, as well as that intermediate step between glycolysis and Krebs cycle where acetyl-CoA was used. FADH2 was another Uber, right, where it also had that process in place. They have these electrons that are high energy. Remember, we said FADH2 was like Lyft. It's just a different company that does the same sort of thing. So these electrons are energized, right? They were stored energized into the sugars of the food, and they're going to continue to be energized. And what they do is they push H pluses across the membrane. And this is on the inner membrane. So this is pushing H pluses from across the inner membrane into the inner membrane space, the space between the outer membrane and the inner membrane. So these H pluses go from the matrix to the intermembrane space, which is between the first and the second membrane. I don't know if that makes perfect sense, but since mitochondria has two membranes, there is a space between membrane 1 and 2, and that's where it pushes it. Now, the H pluses that are pushed into that space get a high concentration. When they come back through, they make ATP. And also the H pluses, when they come back through, they form water and kind of move along their way. You can expect to get about 38 molecules of ATP per glucose. It's really hard to figure out how many molecules of ATP you get per glucose. It's not always consistent. Different things will play a role from it. I mean, lots of things. ATP synthase is the enzyme that the H pluses come through to produce an ATP. It's similar to the ATP synthase that it's in plants, so it's similar in that way, and that's why they both have the same step there. Oxygen then picks up the electrons and the hydrogen ions to make water at the end. And then water is a waste product. Now, I sometimes get the question, well, if water is a waste product, why do we still need to drink water? Well, we don't make enough water to survive off of. So while plants make enough oxygen to survive off of, we don't make enough water. So we need more. And that's it for chapter 4, section 5.